Hello everyone, I hope everyone doing well. So today let's see about gradient. So gradient is nothing but is a proportion that rise and falls of certain factor in the embryo. So the interaction of which brings about the normal development. So the word gradient that derives from the Latin word gradi that means to stop. So for the earlier development of embryo that controls by one or more factors that are called gradients. So the factors that are distributed in a certain proportion of the embryo that rise at the one end and falls at the another end that are called gradient. So the gradient that control the normal development. The next one is types of gradient theory. There are two types of gradient theory. First one is single gradient theory or metabolic axial gradient theory. Second one is double gradient theory. First we see about single gradient theory. This theory was proposed by C.M. Child in 1940. He showed that a development morphogenesis are controlled by a single factor called single gradient or metabolic axial gradient. So the acceleration metabolism is higher at the animal pole, then it gradually decreases towards the vegetal pole along with the median axis. So the existence of the gradient that is proven by the reduction of Janus Green. So this theory says that the head structure development and control by a high metabolic rate, and the tail structure development and control by a due to low metabolic rate. Then double gradient theory. This theory was proposed by Rundstrom and Horstatus in 1928. So this theory was states that the sea urchin X has two factors or two principles. So there are animal gradient and the vegetal gradient. For animal gradient, it has a center of activity at the vegetal pole. The animal gradient diminishes towards the vegetal pole and the vegetal gradient diminishes towards the animal pole. So they overlap throughout the whole leg and they may mutually antagonistic at they interact. For normal development is due to the certain equilibrium between the two gradients. So this system of gradient is called double gradient theory. So according to this theory, normal development depends on the presence of both gradient and an equilibrium between them. So for experimental evidences there are double gradient theory and its mechanism can be provided in two ways. First one is isolation and recombination of blastomers, and the second one is physiochemical methods. So first one is isolation and recombination of blastomers. So this method was proposed by Runson and Horster. So they carried a certain series of experiments in sea urchin embryo to illustrate the existence of the gradient system. Let's we see about. So first experiment number one, the first two cleavages of the meridial and they produce four equal blastomers. When the four blastomers are separated, each blastoma develop into a normal larvae. Then it becomes the blastoma receives equal both animal pole material and vegetal pole material. Then experiment number 2, the third cleavage is equatorial. It produces 8 blastomeres. At the pole of egg, it cuts into an animal half and a vegetal half. So both the halves cannot develop into normal larvae. So they can develop into deductive embryos. So animal pole develop into animal pole structure only that are called animalization embryo, and the animalization embryo consists of an ectodermal vesicle with covering of cilia, and the vegetal half develop into only vegetal pole structure. That embryo is called vegetalized embryo. Then the vegetalized embryo have a large gut without a mouth and a few specicles. They both fail develop into a normal embryos, that is due to the absence of the animal gradient in the vegetal half and the vegetal gradient in the animal half. Then experiment number 3, in 64th cell stages, the embryo has three types of blastomers, namely micromere, macromere and mesomere. They have arranged in 5 tiers. They are as follow. Two tiers are of mesomere situated at the animal pole. They are called animal pole 1 and animal pole 2. Then two tiers of micromeres are situated at the vegetal pole. They are called veg 1 and veg 2. And one more of micromere at the vegetal pole. So these are the macromere, micromere and mesomeres are present in the animal pole and vegetal pole. Then physiochemical experiment. The presence of gradient system, it develops the embryo in demonstrated by physiochemical experiment. Hostetter 1952, he carried out a fantastic experiment on sea urchin blastula. So sea urchin blastula was stained in Janus green, that is another called diethyl saffron azodimethyl aniline. So the stained embryo are placed in a dish sealed with petroleum jelly. Then the free oxygen in the dishes is used by the embryo. After that embryo, respiring using Janus green as a receptor of hydrogen. As the result, Janus green is reduced to a red dye called dimethyl saffron, and then is colorless substance called leucosaffron. So these are the reduction of Janus green in the late blastula of sea urchin. Then the reduction of Janus green follows a very characteristic sequence. The changes of color blue to red in the first seen at the vegetal pole. Then the red color spreads and gradually reaches the equatorial. At the time, the extreme of animal pole that is in blue color that changes into a red and gradually separated towards the equator. When the blue color disappears completely, the red color begins to fade in the same sequence, starting from first form to the vegetal pole. So, the sequence of the color changing is defined owing to the existence of the animal and vegetal gradient. Then Hofstadter is 1955. He separated the animal half and studied the changes in colors. For in the animal half, the animal gradient is predominant and the animalized embryo starts reducing Janus green at the animal pole first, and then the changes in color separated towards the vegetal pole. So there is no changes of color changes in the vegetal pole. Similarly, the vegetal pole is isolated and the vegetal gradient is predominant, and the vegetalized embryo started reducing Janus green at the vegetal pole first, and then the changes in the color separated towards the animal pole. Hence, there is no center of color changes at the animal pole. Then the above experiment clearly indicates that the existence of the gradient that are due to the existence of the metabolic reaction which involves oxidation. So the metabolic rate is higher at the poles and reduced towards the equatorial or in the opposite direction. And see that are the reduction of Janus green in animalized sea urchin embryo diagram is given below. Then this is the reduction of Janus green in a vegetalized embryo. So far we have discussed more about the gradients. That's all. Thank you all.